Good morning, my dear students. Today we are going to complete the blood group trait. As we said in the last lesson, the blood group trait is identified by genetic classification and chemical classification. We talk about the genetic classification of the blood group. We said that the blood group trait is controlled by 3 types of genes: gene A, gene B, and gene O. But each one of us has only 2 of these genes. If we have 2 genes, gene A and gene O, so the blood group will be A, as gene A dominates over gene O. Also, if we have 2 genes, gene B and gene O, also the blood group will be blood group B, as the gene B dominates over the gene O. If we have the 2 genes that determine the blood group, gene A and gene B, so this case is a case of lack of dominance. There is no dominant gene and no recessive gene here. A and B genes are strong. So when they meet together, they produce a new trait which is called AB. It's a case which is called lack of dominance. And the blood group O must contain 2 genes O, as O gene is a recessive, and the recessive trait is always pure. So the individual that has blood group O has 2 O genes. Now we are going to talk about the chemical classification of the blood group. Blood groups are classified into 4 groups: Group A, Group B, Group AB, and Group O, depending on 2 types of substances found on the blood. The first type is called the antigen, and the second type called antibodies. What are the antigens? They are chemical substances found on the surface of red blood cells, while the antibodies are substances found in the blood plasma, and they are antithetic to the antigens. Take care that the antigens are 2 types, antigen A and antigen B. Also the antibodies are 2 types, antibody A and antibody B. Look at the table in purple. In this table, we have the 4 types of blood groups and the types, or the type of antigen, the type of antibody. The blood group A, or the person that has blood group A, has antigen A and also has antibody B. The person that, or who has blood group B, has antigen B and antibody A. As antibody A cannot combine with antigen A because antibody is antithetic to the antigen. So take care that antigen A cannot meet antibody A, or the blood clotting will be happen. About the blood group AB, it contains 2 antigens, antigen A and antigen B, and it doesn't have any antibody. About the blood group O, it doesn't have any antigen, but it contains antibody A and antibody B. About the importance of blood groups. Number 1, it gives the results in determining the enrollment of children to the real parents. So blood groups can be named but cannot prove the parentage. We have life application, page 73 in the book. Please check it. Number 2, from the importance of the blood groups, it determines the process of blood transfusion. So, the blood group character cannot be neglected before the blood transfusion process. It's very important to know the blood group of the donor and the blood group of the receiver. If it is compatible, so the donor can give the blood to the receiver. But if they are not compatible, so the donor cannot give the blood to the receiver. Number 3, blood group trait. The scientists used the blood group trait to study the taxonomy. It's a branch of the biology that study the classification of the living organism. Let's talk about the blood transfusion process. Blood can be transfused between different blood groups according to a specific system due to the presence of antigens and antibodies. Steps of determination the blood group in the lab. How can the technician or the lab technician know the blood group? Number 1, a blood sample is drawn from the person to be determining his blood group. Number 2, 2 drops of blood are placed on clean glass slide. Then we put anti-A on a drop of them and anti-B on the other drop. As we said before, that the antibody is antithetic to its antigen. So you have to know that antigen A with antibody A cause agglutination, and antigen B with antibody B cause agglutination. So if the person has a blood group A, so it has where he has antigen A in his blood. So antigen A with antibody A cause agglutination or positive agglutination, while antigen A with antibody B doesn't cause agglutination. If the person has blood group B, so he has antigen B. Antigen B with antibody B cause agglutination, and no agglutination with antibody A. If the person has blood group AB, so he has 2 antigens, antigen A and antigen B. With antibody A cause agglutination, with antibody B cause agglutination. So if the agglutination happen in both glass slides, so the blood group will be AB. The person that has blood group O, he doesn't have any antigen. So no agglutination with anti-A or anti-B. It's very simple test just to know or to determine the blood group of anyone. This table compare between the 4 types of blood groups. Number 1, or the first point of comparison, the genotype. As we said before, the genotype of blood group A, AA or AO, as gene A dominates over O. So the person that has or who has genotype AO, his blood group will be A, as gene A dominates over the O. About the blood group B, the genotype will be BB or BO, as also the gene B dominates over the gene O, and the gene O is recessive. If the person has blood group AB, so the genotype will be AB, because A and B are 2 strong genes. No one dominates over the other. There is a case of lack of dominance. So the blood group will be AB. The person who has blood group O, it is a recessive trait. So the recessive trait is always pure. So he must have OO genotype, or 2 genes OO. About the types of antigen in each blood group, the person who has blood group A has antigen A. Blood group B has antigen B. Blood group AB has antigens A and B. Blood group O, he doesn't have any antigen. About the antibodies, the person of blood group A has antibody B, and the person of blood group B has antibody A. And the person of blood group AB, he doesn't have any antibody. And the person with blood group O has the 2 antibodies, anti-A and anti-B. Donates to blood group A can donate the blood to A and also can donate the blood to AB because AB is a universal receiver. Why we call the person who with blood group AB is a universal receiver? Because he receives the blood from all blood groups because the blood group AB doesn't have any antibodies. So the blood group B can also donate the blood to blood group B or to AB because AB is a universal receiver. The person of blood group AB can give the blood to AB only, while the person of blood group O can give the blood to all types of blood groups, as we call that blood group O is universal donor because it gives the blood to all blood groups because it doesn't have any antigens. About receives from, blood group A can receive the blood from A or O because O is a general donor. Blood group B can receive the blood from blood group B or O also because O is a general donor. Blood group AB can receive the blood from all types of blood groups because we call that blood group AB is a universal receiver because it doesn't have any antibodies. And the blood group O can take the blood or can receive the blood from just O. About the agglutination. Blood group A has antigen A, so it causes agglutination with anti-A. Blood group B has antigen B, so it causes this agglutination with antibody B. Blood group AB has the 2 types of antigens, so cause agglutination with anti-A and anti-B. Blood group O doesn't have any antigen, so no agglutination with anti-A. In this slide, we said that the blood group O is called universal donor because it donates the blood to all blood groups because it doesn't have any antigen, antigen A or antigen B. While the blood group AB is called the universal receiver because it receives the blood from all blood groups because it doesn't contain any antibodies, antibody A or antibody B.